Hello everyone, thank you so much for tuning in. Again, we're going to be continuing this second exam study video series for microbiology. We're going to be talking about DNA to protein, so this section in particular is going to have a ton of review from other biology classes, but there'll be some new information of microbiology in here as well. So it's going to be a little bit of a review, but then we're going to tailor in some new information that only pertains to the study of microbiology. All right, so a little history. We're going to start talking about Mendel again, Gregor Mendel. And he lived in the 1800s, 1860s actually. This is when he did his famous experiment with the pea plants, and he determined traits were the physical units that were inherited. And the name of those physical units would be genes. It's our first vocab for this section. Later on, two other scientists in 1941, Beadle and Tatum, reported that genes created enzymes. And how they figured this out, they treated mold with x-rays, and there was a metabolic defect in this strain of mold, and this defect was inherited from a single gene. So therefore, we now have the one gene equals one enzyme as the hypothesis. All right, moving on. Let's talk to this slide about some definitions. So, of course, we're going to be talking about the genome, as well as the gene, which we've just stated as the physical unit of inheritance. Then we also have genomics. So with the genome, this is the complete set of genetic information. So this, in the study of bacteria or microbiology, this would be the chromosomes plus the plasmid. Genes will encode a product of a protein, so, and the study of the specific sequence. Sorry about the spelling there. It's late at night. So genomics is the study of the specific sequence. Another fancier field would be bioinformatics, which is a blend of math plus genetics. And according to the professor, you can make up to like 160K just as a starting in that new field. Very surprising and exciting stuff. Make that bread, you know? Alrighty, let's talk about an overview on how the cells must accomplish two tasks in order to multiply. First, the cells must do DNA replication, as well as horizontal gene transfer. This is only seen in bacteria. We cannot do horizontal gene expression. There's also a third way that they could cause variants, which is actually more important than that. In this sense, when we're talking about cells multiplying, we have gene expression. Yes, the horizontal gene transfer is important, but I forgot we're talking about the two tasks for cells to multiply. So we first have DNA replication, and this is basically living things need to replicate. That's the main point here. This is going to be talking about passing that genetic information in the chromosome of the bacteria to the next generation. Gene expression, on the other hand, in order for this genetic info to escape or to be transferred, in order for this genetic info to actually have a purpose to where it exists, it must make a protein. So gene expression is the central dogma, basically, that it's taught. DNA is transcribed into RNA, which is translated into protein. It's, again, a language, a code within biological systems. All right, let's talk very briefly on DNA structure. Again, you basically have a phosphate. You then have your sugar. This is your sugar phosphate backbone. So the sugar is a 5-carbon sugar with only one OH. Because, oh wait, let me think about that. With 2 OHs, I think. I think I wrote them in 2 separate spaces in my notes here. Let me check. Ribose has 2, deoxyribose has 1 OH, and basically you're going to have something that looks like this. So again, a pentagram, or a pentagon, not a pentagram. This is spooky season, but we're not drawing pentagrams in DNA structures here. So again, your carbons, and there is an OH here, and that is important because that's actually the 3'. That's the 3' carbon. And then off of this is carbon number 4, going to go up. This is 5, the 5th carbon. All right, and again, the DNA structure, it's going to be anti-parallel. It's going to be complementary. Each strand will have a 5' and a 3'. The 5' end will have a phosphate. The 3' again will have an OH. There is a sugar phosphate backbone, which we've discussed. It's a double-stranded helix. Hydrogen bonds are between the bases, and we have 2 between adenine and thymine, and we have 3 between C and G. Again, those are going to be hydrogen bonds in between the bases. The hydrogen bonds are there because we need those weak bonds to be broken in order for replication or transcription to take place. When we separate those strands like how I drew in this picture, this is called denaturing the DNA. So kind of like a protein, you could denature a DNA by breaking its structure. RNA, on the other hand, let me switch to a different color. So basically think of the deoxyribonucleic acid, but instead we're going to have ribose. Ribose has 2 of these OHs, like that. And of course uracil is going to be here instead of thymine. Usually RNA is single-stranded, but it could also form into these weird aggregates with itself, depending on the type of RNA you're dealing with. The transcript will quickly separate from DNA once it's finished. The ssRNA, it will bond with itself. That's single-stranded, which we've mentioned. It also has some enzymatic functions. It's also known as the component of the ribosome rRNA, and is also transfer RNA. There's also microRNA and all these other types of RNA, but the main type that we'll be focusing on will be mRNA, or messenger RNA, which is the transcript from the DNA. All right, I think that's all we need to cover there. It's kind of a review again. So 3 types are required for gene expression. We have mRNA, ribosomal RNA, and transfer RNA, which will carry those anticodons since we're in translation. All right, let's talk about regulating gene expression. So we need to regulate it by degrading mRNA transcripts, and this prevents the loss of ATP energy. And I'm going to just put an arrow pointing down because that decreases the chance for that ATP to be wasted. More transcripts will equal more protein. So therefore, if we're going through rapid growth in the cell, this will have this effect. But if we don't need to be growing so rapidly, we don't need all those transcripts. All right, let's talk about the replication of a circular chromosome. And of course, the bacteria have a circular chromosome. So this is usually bidirectional like it is in eukaryotes. And it's also semi-conservative. Again, like in eukaryotes, but the difference being there's a single origin of replication where this process starts. So some proteins will find this origin and they'll bind. First thing that happens generally in this situation, the primases will make RNA primers. And these will be recognized by the polymerases later on. The process of replication also creates 2 replication forks. And just highlight some of the keys here. All right. They meet at a terminating site. And when we draw our picture here, you'll see what I mean by that. So let's say this is our origin and replication down here are the terminating sites. So it's bidirectional. There's going to be 2 replication forks, so they're going to go that way when you're replicating that circular chromosome. This will result in 2 copies. And 1 per daughter. And then if we had this timeline drawing, we're going to again have our origin and replication at the top. Then next, we're going to have replication forks. So the origin there, and then we're going to have the other strand in that chromosome appear like so. Then we're going to have this weird aggregation happening, and this is going to be the terminating site, the center where those 2 meet. Again, our origin and replication, each of those chromosomes has their single origin, and then this will break off, and then if we zoom in, we'll have semi-conservatism after this replication occurred, where one of the strands is acting as a template, and then the polymerase, wherever the primers are landed, it's going to start adding in those complementary bases. Both have a terminating site, and they both have an origin replication. So DNA polymerase, there's 3 types, but in this class specifically, they're just talking about in general. each of the three types of DNA polymerase has their own unique job. DNA polymerase will only work to synthesize 5 to 3, so 5' to 3'. That's the direction where it's going to work. The hydrolysis of ATP will power its function. And the DNA polymerase, sorry, I dropped my pen, it only adds the bases. This is an important piece of information. Usually it's thought that it also puts them together. No, it just adds the bases, again, in that 5 to 3, in that 5 to 3 direction. Let's talk about some of the other proteins that are involved in replication, as well as some important terms that are associated with the process. So we're going to have some proteins again, as promised: gyrase, helicase, the leading strand, lagging strand. Those are, again, strands of DNA. They're not proteins themselves. Here's another protein, though, ligase. Again, ACE, 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 these are enzymes. And we also have RNA polymerase as one of the terms in the next section, not in this section. I'm getting way ahead of myself right now. Okay, let's start back up at gyrase. This will break the DNA strand. To unwind it, helicase unzips to show the templates. There are a ton of other proteins here. There are scaffolding proteins, topoisomerase, all those, but in general, these are going to be the main enzymes involved. But there's a ton of proteins that are recruited whenever replication or transcription are occurring, but these are kind of just the generalization proteins. The leading strand of the DNA is synthesized continuously, while the lagging strand will need these Okazaki fragments. This is another term, and the lagging strand actually goes into 5'3", or the 3' to 5' end. So therefore, this presents a problem, since the polymerase only works or only adds to the 3' end. So therefore, the polymerase will have to do these, again, literally fragment additions onto the lagging strand. So therefore, the polymerase will then replace the RNA primers until it reaches the 5' of another fragment. So again, this is in pieces, not continuously on the lagging strand. Okay, ligase. This creates covalent bonds between the bases of the Okazakis. Alrighty, now let's talk about bacterial transcription. For time purposes, we're going to just call it TRXN. So this again synthesizes 5 to 3. Most of these processes will be 5 to 3 processes. The challenge would be to know where you're going to start on the DNA to transcribe it into RNA. So you need a promoter region, as well as a terminator. Not Arnold Schwarzenegger, no, we need a terminator sequence to stop the transcription because we don't want to go into an area that's out of bounds or off limits on our gene. Those are key terms that we'll be talking about later, so I'm going to highlight them. RNA polymerase doesn't need any primers. And this goes to the promoter. I almost said motorcycle for some reason, and the promoter's upstream of the region that is of interest to transcribe. Okay, so it again ends at the terminator. And how you could tell the direction of transcription is basically just going to follow towards that terminator. So basically, the RNA polymerase is a big fan of Arnold Schwarzenegger's movie, so it's going to go and follow the terminator. There's a little drawing to show the different strands, so 3, 5, and this is 5 to 3. So, this is happening. The strand up here, this is going to be the complement. And then this is going to be the template DNA. The blue. Alrighty. And what's going to happen here is the template DNA is going to be used as the guide, again, hence the name template. So here's our polymerase. It's going to go this way because the terminator is to the right, and we're going to make RNA based off of the template, the blue strand on the bottom. That's going 3 to 5. The RNA is going to be synthesized 5 to 3, and it's going to match the complement strand of the DNA, except for, again, the T's are going to be replaced with U's. So if we had the bases C, G, A, T up on the complement strand of DNA, then on the RNA, the mRNA, it will have the bases C, G, A, U instead, so that T again becomes a U. And of course, since it's matching that complement DNA, it's going to be 5 to 3 left to right. You must be wondering, there's also something called a sigma factor, right? Yes, there's a sigma factor in bacterial transcription, and we'll get into that in a bit. We have some more terms. Again, complement DNA will have the same sequence as mRNA, but mRNA will have U instead of T. We have 2 terms here, monocistronic. And this means 1 gene. But in bacteria, we could also have polycistronic transcription where there's multiple genes. So polycistronic, this is many genes. Going to just highlight those because those are definitions. Okay, let's talk about that sigma factor that we were talking about here. We drew it in pink, so let's write about it in pink. So sigma factor. This is going to find the promoter. It's going to bind to a subunit on the polymerase. And there are different types of sigma. There's a global version, and the global version of a sigma factor, of course, will... Oh, shit. I'm struggling here with the mouse, sorry. this means a certain set of genes are going to be affected via this sigma factor. And archaea as well as eukaryotes are going to have transcription factors instead. So there's a difference between bacteria and archaea, as well as to us as well, because bacteria are the only ones that have sigma units. Okay, now let's talk about bacterial expression. Let me get recalibrated here. Sorry about that. Okay, bacterial expression. And then, of course, this is gene expression. So the promoter will orient the transcription. It will orient the direction, and of course this is found upstream as discussed earlier. Once RNA polymerase has moved past, another RNA polymerase can bind, so this has many cycles. This allows for a rapid and repeated transcription of a single gene. For example, let's say we have 5 to 3 left to right DNA. At the top here, there's a terminator near the 3 end on the right, and then, of course, the complementary strand will be 3 to 5 left to right of DNA. And let's say the terminator is again at the 3' end, which on our other strand of DNA will be on the left side. Okay, so let's have a promoter 1 and a promoter 2 about in the same area on each of the strands, signified by a dot. So we're going to have the RNA polymerase move again towards the terminator. So that means the RNA polymerase will be moving towards the 3 to the left on the bottom strand, and then on the top strand, that is going from 5 to 3 left to right, it's going to move to the right towards the 3 to synthesize. And this allows for, again, the rapid and repeated transition of the same gene. Alrighty. Let's talk about translation very briefly now. Translation will be TSLN. That's again RNA to protein. When we're talking about translation, we're going to be talking about the code, the genetic code. Again, this is where bioinformatics will take place, and we're going to read it as code with math to name the protein. But in general, let's talk about the genetic code in relation to the codon. This is again review. It's going to be the 3 nucleotide bases. The code is, of course, a carrier redundancy. And it's also degenerative. What does this mean? Well, we could have something called the wobble effect, where several codons will still code for the same amino acid, regardless of that third base. The third base, again, in the codon isn't very useful, and therefore, again, we could see this because of the point mutations that could change it, potentially. And therefore, we have a wobble effect. and going into the same realm of redundancy, as well as the degenerative process, the degenerative trait that the codon has. So we have some important codons, such as the start codon. This is AUG. I always forget the stop codon, so there's also a stop, but I can't just remember it right now. This is at the end of the region to be translated. TSLN, region. Alrighty. The binding site for the ribosome is going to be upstream. Alright, let's talk again about the types of ribosomes. So in our bacteria, we have a 70S ribosome, and this is from the 30S and 50S subunit. This is going to be made out of rRNA plus some protein, and it's going to also go 3 to 5. So most of these processes, again, are going to go 5 to 3. Sorry, shoot. I'm so sorry about that, folks. 5 to 3. See, this is what happens when you're very sleepy and you're trying to review notes. Please don't do what I'm doing. 5 to 3. Almost all processes when you're talking about the genetic code are 5 to 3. Okay, so now let's talk about the reading frames. And in order to do that, we're going to also be going into the process of translation itself. So there are 3 reading frames. What is meant by that? Well, there could be a change in the amino acids depending where you start in the code to read all the codons on the mRNA. Again, depends on start. And then you could have something like CUGG, but then if we shift over, we use this reading frame instead, it'll become UGG, and then if we shift once more to we're going to be reading from the G, and that means we're just going to have GG and then the next couple of codons, and each time we're going to be changing that amino acid depending on when we shift the reading frame. Okay, let's talk about tRNA, transfer RNA. This is the type of RNA that actually folds in on itself. This has a region with the anticodon. So if we had the codon UCC, the anti would be AGG. That's what an anticodon is. And it also has an attached amino acid that matches that anticodon. These RNA molecules are recycled in the process of translation. They give specificity and the cytoplasmic enzymes will add the amino acid to it. Alrighty. Here's a fun fact that comes with the translation of the genetic code in bacteria. So translation, let's TSLN, in prokaryotes will actually occur before transcription is ending. So translation before transcription ends. This happens in prokaryotes. So what does that mean? So this allows for efficiency. for incubation. Alright, so what would this look like? Well, we'll have our DNA here, and then a promoter is going again to the terminator sequence. So there's a polymerase. It's going to be making RNA, and then we're going to have ribosomes right on that DNA. Right on that RNA that are making the peptides, as the RNA is still being baled by the polymerase. So it's all happening at the same time. This can't happen in eukaryotes. And the reason why is the ribosomes are in the ER, while the RNA is in the nucleus. So they're just separate, so it can't happen. This translation and transcription at the same time can't happen in eukaryotes because of the spatial difference where they're located. Okay, so now let's just talk about the process in general. I'm just going to go off what I wrote in my actual notes for the class. So there's going to be binding at the ribosome site, and the ribosome is going to find AUG. Again, that's a start codon, and then it'll read every other 3 afterwards. And then in bacteria, we're going to start with something called F-methionine instead of just regular methionine. This is a different form of methionine. That part in the molecule here will be removed. So this version of methionine will be removed once the peptide is finished. Alright, so we're going to have our 5 to 3 again. This time it's mRNA. And in our ribosome, we're going to have, again, 2 subunits. And there are 3 regions in a ribosome. If you're going 5 to 3 left to right, that's going to be the E, the P, and the A site. So what will happen is that the initiating tRNA will bring the altered form of methionine, so the F-met, to the P site. And in that P site, at the start will be AUG. Alright, so what will happen here? Now, the initiating again brings it to the P site. The P site occupied by the tRNA carrying methionine. And another tRNA will recognize the codon in the empty A site. So let's say this codon is CCG. So that's the next codon. What will happen here? Another tRNA that's bringing the next amino acid, which is proline, if you use an amino acid chart, and it'll bring it to that empty A site. There are 2 tRNA currently. An enzyme creates a bond between methionine and proline, and once that bond, a peptide bond, is made, the ribosome will move to the next codon in the 5 to 3 direction. So this will move, let's do another picture here. So pro and then our methionine are going to be the A. Okay, so now let me check. Okay, so now CCG will be in the E site. AUG is out already. This next codon will be UAC, and then let's just say this last one here in the A site that's now there after a translocation will be GAA. Okay, so now our peptide will be on the A site, but it's going to actually move back to the P site. The E site will have the exit, again, the exit site will have the exit of the tRNA that had the proline anticodon, so this tRNA here will be going out. And then another tRNA with the next anticodon and amino acid will enter the A, and then the cycle just repeats. So again, tRNA exits E site, new tRNA occupies the A site, the process repeats. Once the ribosome clears initiating sequences, another ribosome can bind. So another ribo can bind. And this is called a polysome. Termination of this process will be when elongation continues until the stop codon. And I think I wrote it this time, UAG. This is one of the stop codons. And the stop codon UAG isn't recognized by tRNA. So when it's not recognized by tRNA, the enzymes will free the peptide, and the 30S and the 50S subunits will be reused somewhere. They'll be used maybe on another mRNA that's been transcribed. Post-translation in bacteria or in microbes that are prokaryotes is much different than eukaryotic post-translation. So there's going to only be chaperones, which will assist in folding for specifics. And again, proteins have specific structure, so these are going to just be proteins hanging out to help fold the new peptide into a 3D structure. And then there's also going to be a signal sequence. And this is going to be primarily for excretions, or secretion actually, of that protein outside the cell. Okay, so we're going to have this region of hydrophobic amino acids that will be at the end of polypeptides. For exit out through the cytoplasmic membrane. Okay, so if we have our membrane here, again, the polar heads, the tails, yada yada yada, gonna have this kind of a channel looking protein here in the middle. So let's say there's our peptide here and we have some hydrophobic amino acids at the end. So that protein is going to go through this membrane protein. It's going to get that amino acid section at the end clipped off in the cytoplasmic membrane. Actually, no, it's not in the cytoplasmic membrane. Sorry about that. This is going to be out. This is going to be in. Okay, so once we're out, the protein is going to make sure to get into its actual 3D conformation, its specific structure. It might target some molecule here, and since it's probably an enzyme, it's going to break it down into its monomers, and then those could go into the cell. Let's switch gears here and talk about eukaryotic transcription. And we're going to actually have some processing. So we're going to remove introns. So unlike the prokaryotic transcription, eukaryotes will have some processing before translation occurs. So we're going to cap the 5' and add a poly A tail on the 3'. This is going to only be monocistronic. That's, again, one gene is going to be transcribed, and it needs to move towards the ADS ribosomes. Monocistronic was also a vocab term. All right, let's talk about how the process of DNA to protein in bacteria allows them to easily adapt to the environment. So, there are 3 methods. There's transduction, which is one of the 3 methods. I think we're going to start with this one. So, outside info will go inside. This is going to help the bacteria to monitor and react. Version of this would be bacteria here, and it's going to make a signal. And later on, multiple bacteria, maybe the signal says, hey, there's good nutrition here. That's going to just cause more signal molecules to be created, attracting more bacteria. Bacteria are also able to adapt via quorum sensing. This sounds extremely fancy, and it is. So basically, this is density sensing. So this allows for genes useful in critical mass to be realized. Let me scroll down here. So this allows for genes to be activated in critical mass, specifically biofilms or infections, because those biofilms allow the bacteria to adhere and cause those infections. So more signal molecules, this will equal, they're going to get stimulated to make more protections. And you could also use quorum sensing to detect or interfere with the molecules of other species. Again, bacteria are extremely competitive with other bacteria, even ones of their own species too, so they're going to be using quorum sensing to get a feel for the environment, to interfere with those signals of competitors in the space. All right, let's talk about gene regulation. On the other slide, there we go. So operons So on a single RNA, and they have control sequences. So an example would be the lac operon, of course, and we're going to go into that deeply soon, but in general, if there's no lac, let me spell that, let me rewrite the words. I almost said lac none or something like that. So when there's no lactose, the lac operon is off. And this means no enzymes in that pathway will be transcribed and then translated. A regulon, this will allow for global control of several operons. So it's a regulator of a regulator, basically. In layman's terms, I think that's what it would be. Okay. Transcription controls will need to be several things in order to be purposeful. They need to be reversible, as well as it needs to control the number of transcripts. Those are the 2 points that a control needs. So we have an alternate sigma factor. And this will find different sets of genes that aren't found by the regular sigma for polymerase. So again, that's sigma factor, not OF. It's sigma factor. It's not like someone's initials for the polymerase. And then we also have DNA binding proteins. And this is like a repressor or an activator to sequences. For example, the lac operon, we find it, will be repressed. Need a repressor. Alrighty, we also have constitutive enzymes. What these do, they're always on. So these are enzymes that are always going to be synthesized constantly. Usually they are metabolism related. So glycolysis enzymes. I spelled that incorrectly. Sorry about that. I'm running on like 5 hours of sleep, 6 hours of sleep. So these are going to be always on enzymes. These are going to always be synthesized. However, inducible enzymes are of course going to be made only when a substrate is present. So this avoids waste, and then it basically acts as a button to turn on. Then of course we have repressible. And this is routinely activated or synthesized enzymes. One of the examples would be the trip operon, and when growth stops, this pathway is also stopped. Or turned off. Okay, so now let's talk about the lac operon very briefly. So again, if there's no lac, no lactose, this means the repressor blocks the polymerase. No transcription or translation. And the 3 enzymes on this operon aren't dogmaed. So basically they aren't translated or transcribed. But, however, when lac is present, so lac with a big old check mark, this means it could form this different style of lactose called allolactose. And again, allosteric, yes, those are related because this is going to bind to the repressor. This will allow for transcription to be on, and the repressor can't bind. This will then cause a conformation change, and that's basically its mechanism. Please review a video of the lac operon. It's very interesting stuff. Alright, that's going to be everything from this last chapter in this second unit of microbio. The test is Tuesday, so hopefully we got through a lot of things here that could be helpful. Thanks so much for tuning in. Please do something nice for someone, and best wishes to all of you. Thank you again for joining me. Take care. Good night, everyone.